In a laboratory, scientists can isolate a highly effective synthesized chemical compound, but a raw, unstable crystal cannot simply be handed to a patient. There is a vast physical gap between an isolated chemical and a consumer-ready medical product. That isolated chemical is strictly defined as the drug, the active pharmaceutical ingredient, or API. It is the specific molecule responsible for the intended pharmacological effect. Yet a raw API is virtually never administered alone. To be safely consumed, it has to be converted into a functional dosage form, or medicine. That conversion requires the scientific discipline of pharmaceutics. This process relies heavily on excipients. These are the carefully selected additives mixed with the API to ensure the final product is stable, accurate in its dosing, and physically capable of being delivered into the human body. Pharmaceutics, then, is the rigorous scientific and technological engineering required to take a fragile, isolated molecule and build a functional, reliable medicine around it. Building that medicine forces formulation scientists to solve three primary constraints. The first constraint is the physical chemistry of the API itself. Its solubility, melting point, and stability dictate how it can be handled. The second constraint involves biopharmaceutics. The human body is built with biological barriers designed to keep foreign substances out. The medicine must be engineered to bypass these barriers and achieve absorption. The third constraint is therapeutic. The design of the final medicine must account for the specific disease state and the patient's condition, determining whether the dose should act immediately or slowly over 24 hours. To solve these three constraints, scientists rely on a strict sequential pipeline, broken down into 6 distinct stages. Designing a new medicine means moving through this exact chronological blueprint. You cannot begin shaping a tablet without first establishing the fundamental physical chemistry in step 1. Skipping or failing any single step in this pipeline guarantees the failure of the final medical product. We enter Part 1, Physical Pharmaceuticals. Because the vast majority of drugs are administered orally as solid capsules or tablets, they eventually have to dissolve in the gastrointestinal tract. This animation demonstrates that process, visualizing how the degree of ionization and the speed of molecular diffusion control how fast the solid breaks down in a liquid medium. Formulators also have to map the rheology, the flow properties, of liquids and semisolids, alongside the physical behavior of surface interfaces where solids and liquids meet. Every one of these reactions is governed by mathematical kinetics. Establishing the exact mathematical rates of dissolution and chemical degradation is a prerequisite for moving forward. From those chemical calculations, the pipeline transitions into macroscopic physics in Part 2. The physical reality of manufacturing is that most active APIs arrive as crystalline powders. Working with these particulate solids introduces severe adverse macroscopic characteristics. Powders frequently suffer from extreme variations in particle size, leading to separation and poor flow. In large-scale manufacturing, massive tableting machines require perfectly uniform mixing and fast, predictable powder flow. If the powder clumps or separates, the individual pills will have wildly inaccurate doses. Before a single pill can be compressed, the physical chemistry of the molecule and the macroscopic behavior of the powder must be mathematically controlled. With physical properties secured, Part 3 introduces biological threats. Microorganisms must be strictly controlled. This requires a 3-tiered approach: asepsis, avoiding initial contamination, high-pressure sterilization, eliminating present microorganisms, and preservation, preventing microbial growth during storage. Once secured against external microbes, Part 4 addresses how the product interacts with internal biology. This graph tracks the success of internal drug delivery. The y-axis measures drug concentration in the blood plasma, tracked over time on the x-axis. The resulting curve represents bioavailability, the exact amount of the drug that successfully absorbs into the systemic circulation. Formulators use physical properties established in Part 1 to dictate the exact rate of drug release, which directly shapes the arc and duration of this plasma curve. The role of pharmaceutics strictly ends here. The discipline engineers the delivery of the drug to the action site, halting precisely at the point where the body's cellular metabolism takes over. Part 5 is where these scientific threads converge. The first step is pre-formulation, the preliminary testing of how the active drug interacts with potential excipients. This early interaction data acts as a filter, immediately ruling out physically impossible dosage forms for new drug candidates before full-scale manufacturing is attempted. Once a solid form is chosen, powder is processed through granulation, drying, compaction, and coating. The sheer mechanical force requires excipients, fillers for volume, disintegrants for stomach breakdown, binders for cohesion, and lubricants to prevent powder from sticking to the heavy iron machinery. By adjusting these formulation variables, scientists can upgrade a standard rapid-release capsule into a modified release system, reducing a patient's requirement from 3 doses a day down to just 1. Not all drugs survive the digestive tract. Parenteral, or injected formulations, require entirely different large-scale liquid manufacturing protocols to ensure absolute solubility and sterility. Modern biotechnology introduces even steeper delivery challenges. Large macromolecules like insulin and specific nucleic acids are incredibly fragile and degrade rapidly if improperly formulated. To protect vulnerable macromolecules and direct them to specific therapeutic targets, scientists increasingly rely on pharmaceutical nanotechnology, utilizing microscopic carriers like liposomes and polymer drug conjugates. Formulation is the stage where raw data and theory crystallize into a bespoke physical product, meticulously optimized for the specific anatomy it will enter. Part 6 addresses the final reality of the manufacturing process. No pharmaceutical product remains stable indefinitely. By mapping the chemical degradation kinetics calculated earlier in the pipeline, formulators determine the precise rate of decomposition, allowing them to establish a strict, mathematically verified shelf life. To maximize that shelf life, the packaging itself must be engineered alongside the medicine. The pack acts as a vital barrier against physical deterioration, while preventing chemical interactions between the drug and the surrounding materials. Producing a safe, reproducible medicine requires a continuous mastery of physical chemistry, powder mechanics, strict microbiological control, formulation design, and long-term stability testing. Once the final package dosage form achieves regulatory approval, the pharmaceutical technologist's role in the pipeline is complete. The product undergoes a final handoff, transitioning out of the laboratory and into the realm of dispensing, patient interaction, and clinical pharmacy practice. Pharmaceutics provides the highly disciplined mechanical and chemical bridge that guarantees a fragile molecular discovery can survive the physical world